So our objective is to understand the role of enzymes, specifically in chemical reactions, and talk about how that is regulated later on. We're going to talk about how enzyme activity is then regulated. So let's first start by talking about what an enzyme is. So we know it's a protein. It's also a catalyst for reactions in biological systems. So enzymes basically make reactions go faster. They speed up the rate of the reaction, and they do so by decreasing the activation energy for a specific chemical reaction. And what that means is that they lower the amount of energy that's required for the reaction to take place. They make the reaction more favorable, more likely, and they increase the speed of the reaction as well, okay? Enzymes bind to a reactor. We can also call that a substrate. That gives us an enzyme substrate complex. And then from there, they then spit out product on the other side of these bidirectional arrows, okay? So the enzyme substrate, we've got a bidirectional arrow here, gives us an enzyme substrate complex, and then that in turn, again, via a bidirectional arrow, leads us to product, and then we can spit the enzyme out on the other end. So essentially, these bidirectional arrows are telling us that this can proceed in the forward direction to give us product along with our enzyme, or depending on the product concentration, we can also proceed in the reverse direction, which gives us substrate and then the enzyme as well. Okay, I hope that's clear. And then in the middle here is kind of the enzyme substrate complex as it can proceed in either direction. So let's draw here an example of how enzymes actually work. Let's talk about the activation energy barrier. Okay, so let's look at this graph, and we're going to put the energy that's required for this reaction on the y-axis. That's going to be in kilocalories. And then on the x, we're going to put the time that it takes for the reaction to proceed. And that'll be in seconds or so. So let's start off with our reactants here. Oops. All right, so we've got reactants here to start. And so when the reaction proceeds without the enzyme, this is how much energy it requires, how much time it's going to take, and then eventually we'll get to products on this end. Okay. And so from our baseline, the baseline here is representing where the energy requirements are when we are at reactants. So we can just set this as a baseline in terms of the energy requirements. And we can see how, as the reaction proceeds, we require much more energy until eventually we get to our products here on the right. So this is how the reaction is proceeding without an enzyme, right? So let's kind of just label that up here. Oops. Okay. On the other hand, let's look at what happens when our enzymes are involved. So let's make another, another curve here, and this is going to be with an enzyme. So how will the reaction proceed in the presence of an enzyme? Okay. So from reactants, we start over here again, and this is the energetic requirements. But then what we see is that we don't need as much energy. Also, the reaction proceeds much quicker. And in terms of time and energetic demands, we get to our products much sooner, okay? So this is the activation energy required for this reaction. And then this is the activation energy required for this reaction, okay? And we can see how that's almost double, right? When we have an enzyme, the reaction takes place much faster and we don't need as much energy in order for the reaction to proceed, okay? And so let's kind of just sum up this with a few descriptions here. So one, we can say that the enzymes lower the activation energy required. And two, we can say enzymes speed up the rate of the reaction. Okay. Okay. So this is what enzymes do. These are the two characteristics of enzymes in terms of how they affect the rate of a reaction. And so we've got lots of enzymes in our body. We'll talk about some examples later on, and we'll talk about how they actually help reactions go faster. So for example, let's think about an enzyme, DNA polymerase. So DNA polymerase helps to synthesize strands of DNA by adding those nucleotides. And so they actually help that process to go faster than if we had to do that without the presence of an enzyme, okay? All right, so let me pause here and see if there are any questions, and then we're gonna keep going here. Okay. All right, so let's keep going here. So here's what we just kind of drew. So again, just to recap this, the activation energy without the enzyme is much higher. That's how much energy is required for the reaction to proceed. And then in the presence of the enzyme, that's significantly lowered. We get to our products quicker and we need less energy to do so. So it makes things in the body much more efficient than if enzymes were not present. So now let's talk about the specificity of an enzyme to the substrate or the reactant. So there are two models that explain how an enzyme is specific to a specific substrate or reactant. The first one is called the lock and key model. So the lock and key model says that an enzyme is like a lock and it has a specific fit. The same way a specific key would fit to a lock, same way the substrate is specific for that enzyme. Now there's a problem with this model in that it doesn't explain the reversibility or the bidirectionality of most enzymes. So for example, we're saying that this is enzyme E. It's got an active site where the substrate will bind. And so it has a specific fit that will only allow one type of substrate. As we can see here, it's bounded in its shape, and so it's only going to fit substrate C. So substrate C binds, we have the reaction proceed in the forward direction, and then we end up with product and enzyme on the other end. And this is where the problem comes in, is that it doesn't explain how the product now can bind to that enzyme once again and give a substrate on the other end. So if there's an induced fit of the substrate to the enzyme, that doesn't explain how the product can also bind to the enzyme. So there's kind of a problem with this first model of substrate specificity. The second model here is induced fit model. And this model, or this theory, suggests that an enzyme will come in contact with a specific substrate. It will then manipulate itself to fit that substrate. The reaction will proceed, and then it will give product and enzyme on the other end. Okay, and so this does explain the reversibility, right? Because now the enzyme is kind of shape-shifting itself to accommodate the substrate on one end, and then it will shape-shift itself to accommodate the product on the other end, okay? So this second model, the induced fit model, is much more favorable. It actually explains the bidirectionality or the reversibility of the enzyme in terms of the reaction, okay? All right, let's talk about some of the other players involved in enzymatic reactions. So we have two types of components that can bind to an enzyme. We've got cofactors. Cofactors are pretty much directly involved in the enzymatic reaction. They're non-protein components. They can be trace metals, so things like magnesium, copper, zinc, iron, and they kind of basically help the enzyme to hold its function or hold its conformation. So they're directly involved in the catalytic reaction of the enzyme. They also help to stabilize the enzyme substrate complex as well. On the other hand, we've got coenzymes. So coenzymes have no catalytic activity, but they're just kind of supporting structures. They help the enzyme to function properly, and they participate directly in the enzyme reaction. They usually derive from vitamins. They can also be NADH, so carrier molecules like NADH. We talked about those nucleotide-derived molecules, FAD being another one, that help to support the reaction, but are not necessarily involved in the catalytic mechanism of the enzyme. And to kind of make this a little bit simpler and give you some real-world examples as to how these two differ, I like to think of the cofactors as being the steering wheel in a car, and the coenzymes as being maybe the headrest or the handrest, right? The headrest and handrest make the experience more comfortable. They support the driver, but they're not necessary for the actual functioning of the car, okay? Cofactors, on the other hand, would be comparable to the steering wheel, right? We can't actually drive the car without the steering wheel involved. So coenzymes would be like the headrest or handrest. Cofactors would be actually involved in the process, like the steering wheel, okay? Let's see here. So I've got a question. Someone is asking, what is catalytic activity? Okay, so catalytic activity is just the activity of the enzyme. The action of the enzyme in speeding up the reaction. Now, it can be specific to whatever enzyme we're speaking about. There are many types, but it's just the action of the enzyme in speeding up the reaction. And that will go faster with an enzyme. And so the coenzyme, we said, is not involved in the enzyme's activity, whereas the cofactor is more intimately involved in the enzyme's activity, okay? Let me pause here again and see if there are any questions about this, any further questions about this. So again, think about this in terms of the steering wheel versus the headrest or the handrest in a car. And think about what is really involved in the enzyme's work versus what is just supporting the enzyme in its function, okay? An example of a cofactor would be magnesium. So magnesium happens to be a trace metal that's required for DNA polymerase activity, that enzyme that helps us synthesize DNA as well. So the synthesis of DNA can take place without magnesium, but it's just much better, it's just much smoother, it's just much more supported with the presence of magnesium. Okay. All right. Now, in terms of the rate of the reaction, let's talk about some factors that affect how fast the reaction goes in the presence of an enzyme. So an enzyme's catalytic rate is how many molecules are generated per unit time. So that is going to be affected by the substrate concentration for one. So if we have lots of substrate, the enzymatic reaction is going to proceed much faster. If we have a little bit of substrate, the enzymatic reaction will proceed much slower. Another factor is the enzyme concentration. Obviously, if you've got a lot of enzyme floating around, we're going to have a faster rate. If we have less enzyme floating around, we're going to have a slower rate of the reaction. The affinity of the enzyme for the substrate. So the affinity is basically the affiliation of the enzyme and the substrate, how attracted they are to each other, how likely they are to engage or to bind. And so if the affinity is really strong, if they really want to interact or bind with each other, we're going to have a faster rate of reaction. Whereas if the affinity is weak and these two subjects, the enzyme and the substrate, do not want to interact with each other, we're going to have a slower rate of the reaction. And then finally, the temperature and the pH. So we kind of talked about this. In a specific temperature, we can denature our enzymes. Remember, enzymes are simply proteins at the core. And so if they're exposed to higher temperatures, they can become denatured. If they're exposed to acidic pHs, they can become denatured as well. And so that can influence the rate. But also just generally, in a warmer, slightly warmer temperature, the reaction will go faster as well. It's going to increase the rate of the reaction up to a point where you could possibly denature the enzyme. So a warmer temperature will speed up the reaction to a threshold where you could possibly denature the enzyme. And so these factors, temperature and pH, could definitely affect the rate of the reaction, or the rate of the enzyme in the reaction. Okay, and just to reiterate here, the catalytic rate is how many molecules are generated in time. So as we're going from either substrate to product or product back to substrate, how much of that substrate or product are we producing per unit time?